This is happening. All right. Good afternoon and welcome to the High Fidelity Seminar Series. It's a really great pleasure to introduce today's speaker. Dr. Jayme Dahlin is a physician scientist currently working in the pharmaceutical industry. After earning his MD and PhD degrees from the Mayo Clinic, he completed a residency in clinical pathology at Brigham and Women's Hospital and Harvard Medical School. Dr. Dahlin then completed a postdoctoral fellowship in chemical biology at the Broad Institute, where he studied the effects of nuisance compounds and complex cellular assays in the laboratories of Dr. Stuart Schreiber and Dr. Bridget Wagner. After completing his training, he performed chemical biology research at the National Center for Advancing Translational Sciences within the National Institutes of Health before transitioning to industry. Dr. Dahlin is board certified in clinical pathology from the American Board of Pathology and currently serves on the scientific editorial boards of the NIH Assay Guidance Manual and the Chemical Probes Portal. He has authored more than 60 peer-reviewed publications in the fields of early drug discovery, nuisance compounds, and assay development. And today he will be sharing his insights about the impacts of nuisance compounds on cellular assays. It's a virtual platform, but please join me in welcoming Jayme. Jayme, it's a great pleasure to have you here. And as agreed, I'll be happy to drive your slides. So just let me know.
Click.
You have the floor.
Yeah. All right. What I want to talk to you today about are nuisance compounds in cellular assays. So Nathan, can you click? All right. So I have my contact info if you have any follow-up questions. Just some disclosure here. I'm a current employee of Agios Pharmaceuticals, but this work was done at the Broad Institute at NCATS, and I'm speaking in personal capacity, so views my own. Next slide. And so today I thought there'd be two objectives, what I'm hoping to talk to you about today. And the first is we can review some of the chemical mechanisms of cellular nuisance compounds. And the second is to review some best practices for how to identify and mitigate their impact in drug discovery and chemical biology. Next. And so I thought I would do this with two illustrative cases. Click. So the first example is going to be from some postdoctoral work at Broad Institute, where we use this assay, probably a lot of folks have heard about, called cell painting, to see, to kind of measure the effects of nuisance compounds on cellular readouts. And then the second, next, is an example from SARS-CoV-2 drug repurposing. In this particular case, we'll be talking about some previously not, this is the first time we're disclosing this publicly, with NCAT's blessing, of course, about the preclinical effects of Ivermectin. Next slide. And so just by nature of having this talk, we know that nuisance compounds are a significant problem in drug discovery. So next. And so the example I like to give of why this is so important is let's contrast these two molecules. The first in the blue on the right, artemisinin, this is an anti-malarial compound that its discovery was, was awarded a Nobel Prize some years ago. And you can see there's a steady number of publications, but let's contrast that with the molecule on the left and the points in the red, curcumin, which is a natural product that the NIH has spent over $150 million as of 2017. It's been the subject of an exponential amount of publications. And I wouldn't even want to know how many are in like 2023, probably over 3,000 per year. And there's not a single FDA approved indication for this. And yet it's been in clinical trials for basically, you could say almost everything. And so, well, there are many more examples. I think the point should be taken that a significant amount of scientific resources are being used on compounds nuisance compounds, which we define as having limited utility as tools or drugs. Next. And so this isn't just a problem for academics. It also affects industry. I can, there's examples in there, and I'm talking to NIH. NIH is not immune to publishing nuisance compounds, sorry to say, but it's true. But there's also, it's understandable because it's really hard to understand what a nuisance compound is and how it behaves in cellular assays, because there's thousands of targets, there's complex interactions, there's even compartments within cells, there's metabolism going on, and a lot of these assays are expensive and difficult to do. So it's a hard problem. So next slide. And so what do we talk about as a nuisance compound? What is it? Well, I think we can define it as chemicals or compounds that look like they are bioactive and are going to be the next chemical tool or drug lead, but actually fall far short of that and are just going to waste your time. And how I think about it in others in the context of cellular assays is we have one large bin in blue here, technology-related interferences. And you can think of these as compounds that interfere with the assay technology. They don't actually change the underlying biology of your system. Examples would be like a colored compound in a colorimetric assay, or a compound that fluoresces in the GFP channel, and you're doing a GFP reporter assay. Contrast that with this yellow and gray bin here, where what we call non-technology related interferences. These types of nuisance compounds will actually modulate the biology that you're studying, but they do so not by interfering with the technology, but through mechanisms that are non-specific or very hard to optimize and to translate into something that's actually going to be useful as a tool or a drug. And in the yellow, there's certain mechanisms that usually aren't associated with one particular target. They're kind of pan-active, and these are non-specific electrophiles, aggregators, surfactants, things like soaps that just kind of lyse your cells, oxidants or chelators. Then there are other compounds that are more nuisance mechanisms that are related more to certain targets or pathways. And a lot of what you'll see in cellular assays are cytoskeletal poisons. I mean, they're a great basis for some drugs, but we're not necessarily looking for those all the time. Mitochondrial poisons, as we found out, Brian Choiket's work from a few years ago, things like hydroxychloroquine in SARS-CoV-2 repurposing assays, there's a lot of these lysosomotropic agents that will induce drug-induced phospholipidosis. That's actually not an optimizable mechanism. And then what I want to point out here in the pink is that there are occasionally high-quality cell-active hits that can interfere with the technology, can have some cytotoxicity, but generally we avoid those, especially early on. And of course, we're speaking to NCI, so there are cases where you want some cytotoxicity if you're making, let's say, an anti-neoplastic, but you want it to be selective, right? So that's where you can kind of contrast cytotoxicity with nuisance compounds versus drugs and other useful probes. So next slide. And so I thought I'd share some of the top misconceptions that I hear in rebuttal letters when I review papers or just presentations. So click. The first is my compound has a positive cellular phenotype. Well, just by definition, these nuisance compounds, they're known to produce positive readouts. So just because you have a readout that's positive, that doesn't necessarily mean you're in the clear. Next. Next is, well, it's an FDA approved drug is my hit. So that can't be, it's a drug. Well, that's especially important, as I'll show later on, that if you use a particular FDA approved drug outside of kind of its normal intended use, funny things can happen. Next. Another is appealing to the fact that we see activity in an orthogonal assay. Well, you could have, you need to see activity in an orthogonal assay, right? That's important. But some of these non-technology interference compounds, by definition, they are going to be active and they're changing the biology. So it's just not an intractable way. So you can have a nuisance compound be active no matter which way you analyze it. Next. There's an appeal to reproducibility. Well, these nuisance behaviors are often related to the chemical properties of the compound, and those are highly reproducible. So guess what? The readouts are often highly reproducible. Next. And you'll see here an example later on, this appeal to, well, are compounds, let's say, not toxic at the concentrations where we're seeing the activity? But as I'll show you, kind of depending on how you set up your experiment, you might be, that might not be an accurate statement. And then finally, next, yep, so nuisance compounds only happen to other folks, and that's simply not true, okay? Next, so when I first wanted to analyze this as a postdoc, you probably heard about this assay cell painting, and I'll talk about that in the next slide. But one thing we did is at Broad, before I came, click, they profiled 30,000 compounds at Broad in the cell painting assay, click. And what you'll notice here, highlighted in the red, is the most significant morphologies, or how cells appeared in this assay, were most often caused by compounds that also reduce the number of observed cells. So that's that enrichment there in the upper left corner. And that's not surprising. Click. So this is a schema that's been shown by a lot of folks at Pfizer, Tim Foley, Fabian Vincent, and others. And I think it's a good concept to really understand. So if you have a cellular phenotype shown there on the right, and if you're measuring a pathway that starts from a stimulus and goes through all these biological nodes, whether that's a binding event or a post-translational modification, different things will ultimately produce the phenotype. Now, there can be cases where in the red, where it's a non-desirable mechanism of action that you have to sort out. And then on kind of the bottom right of the cell, you can see there are cases where something completely unrelated to the biology you're interested in can produce the same phenotype. And so you have to kind of think of as a cellular readout as a black box that's really dependent on, what your assay is capable of measuring. And another way to think about it is if you have a loss of signal or a readout that's dependent on disrupting a cellular process, remember the analogy I think I've read somewhere is that it's a lot easier to make a car, break a car and make it go slower than it is to improve it or soup it up. So think of that, and we're going to show you some examples. So next slide. So I mentioned cell painting. This assay actually is being used a lot by pharma, academics, and even government now. And it's actually a very simple assay. You can simply take cells, any cells, we use U2OS, and you add compounds, you incubate them, and then after your 24, 48 hours, you then add a cocktail of stains that will, in this case, target things like the nucleus, the plasma membrane, mitochondria, et cetera. You stain, wash, and then you image all those cells in all these different spectral channels. And then you essentially can, with software, get for every cell over 1,000 features per cell across all the compounds. And then you can use big data approaches to try to look for patterns between the cellular features by morphology and bioactivity. And the basis for this work I want to talk about in the first part is, can we use cell painting and unbiased morphology profiling to identify phenotypic signatures associated with nuisance compounds? And the answer, of course, is yes. Next. And so we profiled at Broad 200 cytotoxins and nuisance compounds across multiple concentrations in cell painting. And we also did a host of companion cellular health assays. And what we found is when you analyze all those features, you crunch the numbers, you do a principal component analysis, for example, that's on the left here. And we basically ask, can we cluster the behavior of these compounds in cell painting by their phenotype? And when we look at have our analysis find 9 clusters, for example, we see that several of these prototypical cytotoxic chemotypes like kinase inhibitors, genotoxins, tubule inhibitors produce distinct morphological clusters. But was striking and not at all surprising when you really think about it, the most feature space was actually taken up by the cytotoxic and nuisance compounds that the only pattern that you were really able to find was that they were associated with gross cellular injury by some of our companion assays. And so really what that shows is that it's very easy in some ways to generate a signal in assays like cell painting or high content screening. You can infer by compounds that just damage cells. And this isn't surprising for those of us that do this, right? So next slide. So an example, like with real compounds, is there's a very big interest, right, in targeted covalent inhibitors. But there's also kind of the fear is, what about if your compound's reacting with everything? And so cell painting, we were able to kind of compare and contrast the behavior of nonspecific electrophiles shown in red versus targeted ones shown in blue. So the upper left is compound 144. You can see as you increase the concentration, the kind of the open boxed ones, the activity score in cell painting is increasing, and that's mirrored by inverse decrease in the amount of the cell number. And then you can see as you go, you increase the concentration, its cluster is going from cluster 1 at the lowest concentrations all the way to cluster 9 at its highest concentrations, which is the cluster associated with gross cellular injury. And there's an example picture. When you look at an inactive analog, this 149, it's essentially no different than DMSO. So those cells on the upper right, that's what essentially your controls should look like. Now, if you look at these targeted covalent inhibitors, not surprising, a lot of them are non-toxic, except at concentrations that grossly exceed their EC50s for their intended uses. And so already you can see that you have non-specific or nuisance electrophiles that can produce profound readouts, and yet compounds that, and then really these don't really do anything useful, versus compounds that are useful in other contexts, but actually don't do anything really in cell painting. And so that's, an important takeaway right there. So next slide. Then going back to my grad school days, I studied a lot of the compound class called lysine acetyltransferase inhibitors that are important for epigenetic writing. And so at the time, we found there's about 20 of these compounds published as CAT inhibitors, but as you can see here, almost all of them, and we showed this in a previous paper, that almost all of these compounds that were published were non-specific electrophiles or aggregators, and essentially were classical nuisance compounds. But in the recent years, there have been some folks from AbbVie and Jonathan Bale and Monash University who have published high quality, what we call next generation CAT inhibitors that essentially check all the boxes for a quality probe. And what we wanted to see with the cell painting is, can this give us a clue about quality versus low quality probes? And the answer, not surprisingly, is yes, because if it's related to cellular health, we're able to see that in cell painting, where on the plot here, you can see the blue are essentially clustered around where DMSO would be in the middle at 0 and 0. But then you see in the kind of bottom left corner, there's all these bigger red dots. Those are these historical CAT inhibitors that are essentially toxic. They're showing the gross injury phenotype at the concentrations that they were purported to be active in the original reports. So click. So again, just to show you further data on that, you can see in the top row, these are these next generation CAT inhibitors where they don't perturb the number of cells in our system, and they're kind of very weakly active. And we have Western blot data, for example, to show that they are on target and working. They're just not producing a phenotype. Contrast that with these low quality CAT inhibitors where they're producing a significant cell painting phenotype, and some are showing by Western blot no activity, some are showing some activity, but it all comes down to being able to just inducing cellular injury. And one thing you'll notice here in the bottom left, some of these compounds that are aggregators, they don't really do anything in this assay, then once you kind of approach the critical aggregation concentration, they start to show significant activity in cell painting. So it really kind of fits with what we know about nuisance compounds. So next. And I mentioned, how do we know that that cluster is associated with cellular injury? Well, one, we purposely pick cytotoxins and nuisance compounds, but we essentially profiled all of them in concentration response live cell imaging, multiplexed imaging. And what we found here, if you look on the right, we measured them for essentially 3 days under conditions that were mimicking cell painting. The compounds with the highest activity of cell painting also were the ones that had the lowest confluence, or the fewest number of cells, which fits with the data I showed you earlier. And that also, if you look at the bottom right plot, compounds with the fewest cell number actually had, again, the fewest with the confluence. And again, in pink, cluster 9 is the gross cellular injury. So really, again, click. Kind of summarize this cell painting work, click, is one, significant cell painting morphologies can rather easily be caused by nuisance and cytotoxic compounds. Click. These cell painting injury phenotypes, they correlate with live cell imaging cellular health assays. And finally, they you can then, it follows that cellular health assays are going to be very high yield experiments for assessing cellular nuisance behaviors, especially if you're just doing some unbiased profiling in cell painting. One of the first things you should follow up with is not just the relative cell number, but doing some of these kind of companion assays. And so that takes us to the second part, where this is talk about some work we did at NCAS to kind of show you an example of how a nuisance compound can creep up and have significant effects. And so click. So we all should have heard of ivermectin as it relates to SARS-CoV-2. And just to kind of refresh your memory, what happened was in 2020, there was this one figure paper published where a group treated Vero cells with SARS-CoV-2, and after several days, they noticed this significant decrease in viral RNA. Kind of one thing led to another, and all of a sudden, ivermectin was being used worldwide off-label for as a potential or purported antiviral compound. And obviously, we all know that some people feel very passionately one way or the other about this. And that's one thing we wanted to work up at NCATS at the time was, is there anything we can add from a preclinical perspective? So analyzing this, I think it's important to go back. Why did this group pick ivermectin in the first place? Click. Well, the answer is about 10 years ago, there was an AlphaScreen performed looking for antivirals. And the premise was this, that this group was looking for disruptors of a protein-protein interaction between a viral nuclear localization signal and a nuclear import machinery. The idea being, if you could disrupt that interaction with a compound, you're not going to get the viral proteins into the nucleus, and then you're going to have antiviral effect. And they performed a AlphaScreen on a fraction of the low-pack, so one of the 4 plates of low-pack at one concentration, and then some associated data after that. And that's led to, click, a model shown here for SARS-CoV-2, but also been invoked for other viruses, where ivermectin will disrupt a viral protein nuclear localization signal and this important alpha beta 1 complex, which then leads to a supposed antiviral response. And basically, all of this has led to hundreds of clinical trials. And if you're like, well, I mentioned before, nuisance compounds don't just happen to other people, they happen to us. If you pay tax dollars, you should know that, click, and click. The NIH even funded 2 randomized placebo-controlled studies with over 1,000 patients each, testing the effect of ivermectin versus SARS-CoV-2 or COVID-19, and not consistent with other studies of similar scale and quality, they saw no effect. And so at the time this is all going on, we wanted to ask what is there potentially some explanation we can offer the preclinical models. So click. The first thing I wanted to look at was how does, let's go back to the original screen. Can that tell us answers about what's going on? Well, first, click just a refresher on what AlphaScreen is. And the way that works is you have a donor bead that's excited with a high wavelength laser with some chemistry inside the bead that produces an excited form of molecular oxygen, singlet oxygen, which then can diffuse out. And if it's in sufficient proximity to an acceptor bead, it can diffuse in that acceptor bead, additional chemistry is performed, and that generates lower wavelength signal of light that can be detected. Now, you can make a PPI assay by essentially coating one protein with biotin, another with incorporating a His6 tag, adding capture reagents to the donor and acceptor beads. And if everything works as shown here, you form this proximal complex that can generate your signal. The idea being if you have a compound that disrupts that protein-protein interaction, the beads will be too far apart and you don't generate a signal. So click. Knowing that ivermectin is not very soluble, one of the first things we did was just look for evidence of solubility. And when you use the same conditions that's used in the AlphaScreen, you find that the ivermectin produces or absorbs light, significant amounts of light at the excitation and emission wavelength shown there, the dashed lines. And that when you add tween 20, just a small amount of detergent, that absorbance completely decreases. So that suggests there's incompletely dissolved particles of ivermectin. And that's important because it may lead to interference. So we'll use some different experiments. Click. The first on the left is instead of using the proteins used in the original assay, we used a biotinylated His6 peptide. And without using any of the proteins from this original screen done 10 years ago, just with this control reagent, using the otherwise identical conditions, you're able to generate an almost exactly identical concentration response curve, including the potency and partial efficacy. You'll notice there that you have kind of a max efficacy of about 75%. That's, if you were to go back to the previous slide, you would see that that's almost the exact same curve. Believe me? Okay. So thanks, Nate. Then if you also use true hits, which it's a set of biotinylate or streptavidin-coated donor beads and biotinylated acceptor beads that you put together. So essentially you don't need any of that proteins in the middle. It forms this complex without the other proteins. You can get the same exact signal. And then finally, we used another control reagent called omni beads, which is essentially all of the alpha chemistry within one bead. So it's one type of bead floating. And when you add ivermectin, the signal does not change. And click. As shown in this assay guidance manual chapter on kind of AlphaScreen interference, our logic led us to hypothesize that ivermectin is interfering with the AlphaScreen assay by quenching that singlet oxygen transmission. And why do we think that? Well, it doesn't change the signal in the omni beads, because all the singlet oxygen itself is within the omni bead. But when it has to diffuse between a donor and acceptor beads in the kind of the two left control reagents, then it's able to be, for example, physically quenched by these insoluble ivermectin particles. But we're talking about cellular nuisance behavior. So click. One of the things that we did was we first took, we did a SARS-CoV-2 antiviral assay where we took A549 cells and we used Fluke labeled SARS-CoV-2 virus, where we're able to quantify viral burden by luminescence. And you can see here in the red, 3 different independent samples from different vendors of ivermectin produce basically almost identically, data to what's been previously reported. So that's great. But we also noticed that when we did a cell titer glow counter screen, it almost exactly paralleled that of the antiviral. But what's interesting is that in the original reports on ivermectin and antiviral, they either say that there was no cytotoxicity observed or only at higher concentrations. So that prompted us to kind of investigate this further. Next slide. So we did what I call a promega panel. We used a lot of different promega assays. So the first was looking at cellular viability with 3 different orthogonal methods. And one of the things I really want to advocate here is the importance of profiling cellular health across multiple concentrations and multiple time points, including those that are longer than your primary assay. And what you'll find here is these are all kind of internally consistent. But you can see here that once you go to 24 and beyond hours of ivermectin treatment, the host cell viability significantly decreases, right? But you can tell that if you look at cell titer glow versus Resazurin, depending, on, what time point you're using, you might be led to kind of different conclusions about toxicity early on. So that's why, click. I'm a strong advocate of also looking at other, not just looking at cell titer glow, but other methods of cellular health. Because what's striking is when you looked at caspase 3-7 activation, just at 6 hours, you're able to see almost a 20-fold increase in activated caspase 3-7 by ivermectin. And if you were to go look at that same cellular viability at 6 hours, you would not see that at those same concentrations. But you already seen evidence of gross cellular damage by this particular assay. And again, that's similar when you do a membrane integrity assay like AlphaScreen. So when you can put this all together, I hope this shows the importance of, one, testing multiple concentrations and testing at multiple time points and even by multiple methods. They all are interrelated and they can kind of help you build a complete picture. So next. And one of the things, we talk about is structure activity relationships. And that's important because what you're looking for early on in discovery is SAR, because that provides evidence that this compound or lead can be optimized by medicinal chemistry. If not, that this basically what you see is all you can get, right? And a hallmark of a lot of these nuisance compounds is you see a flat SAR. Now, click. We took a library of commercially available ivermectin analogs, and there was almost in the same F-Fluke assay, we see almost completely flat SAR with one notable exception, which might be due to solubility, I believe. And so this is already, suggestive of something that's not optimizable. And again, it's highly reproducible. But I also would want to caution you about the opposite approach, click, where not shown here, but there are other cases where you can have an apparent structure activity relationship, but actually that reflects an undesirable mechanism of action. So we have examples in cell-free assays where you see an SAR, but actually that's all completely explainable by the ability of a compound to form aggregates or the ability of a compound to react with thiols. So it can go both ways. Next. So one of the things, if you're down to a few compounds, selectivity profiling, it can cost a lot of resources, but if you're down to a few compounds, it's well worth it, in my opinion. And you can use some of your earlier data to generate, to pick a non-specificity panel to help home in on a mechanism. Let me show you an example. Click. With ivermectin, we know that its normal indication, its anti-parasitic activity, it's going to target these ion channels, right? And the way it does it is it binds in the membrane-facing portion of the molecule, as shown here in the inset, right? So that already and that's at like 10 nanomolar. So that tells us something. That tells us that this compound really likes to go to membranes. So that's an important clue. Click. So what about if a compound's affecting membranes? That would explain the cytotoxicity we're observing. So let's create a panel to look at non-specificity. And so we did that 2 ways. The first, click, is we had it profiled by the psychedelic drug screening profile at UNC and Brian Roth's group. And they found, and one reason for this was you saw potentially a lot of neurotoxicity that was being associated with high dose ivermectin poisoning. And so a lot of these are, that was one rationale for first looking at that too. And he found that over a third of the targets profiled, ivermectin significantly inhibited these GPCRs that are completely unrelated to the anti-parasitic target and also the antiviral purported target. Click. Then we also did an orthogonal beta arrestin cellular assay where they profiled over 150 GPCRs. And similarly, almost a third of the GPCRs profiled were significantly inhibited by ivermectin. Click. So what do these have in common? They all have, these assays all include membranes. These purified GPCRs, they're enclosed within the membranes, and same with these cellular assays. Click. And that leads, to, I want to talk about probably one of the more underappreciated sources of cellular nuisance behavior. And this was really a lot of work done by my collaborator, Olaf Anderson at Cornell. And his basically made a career out of this. Click. So you can imagine in a membrane-based target, let's say this is an ion channel on the left, there exists an equilibrium between an off state and the upper left. And when the membrane is bent or compressed, that can alter the dynamics of the protein and move it into more an active state where ions can flow through. Now, what Olaf has studied are drugs that will incorporate into the membrane bilayer. They can alter the physics of the membrane. They can bend and compress it and then shift that equilibrium to an open state. Okay, click. So alteration of the bilayer structure can obviously perturb membrane-based targets, but also all the downstream targets associated with those. So click. How big of a problem is this? Well, when Olaf looked at the pathogen box, and he used this assay I'll talk about on the next slide, you can see in red a significant amount of those compounds significantly perturbed membrane bilayers, and these I can tell you were most associated with cytotoxicity. Next. Next. So we used, because we're thinking now ivermectin might be perturbing the bilayer, that would be consistent with this antiviral activity that we're seeing, this cellular damage, especially that's immediate, right? And so the way this assay works is you have some special vesicles that are loaded with this fluorophore, ANTS. And then on the outside of the vesicles, you have thallium ions. And there's going to be kind of a natural rate of these thallium ions to get into the cell where they can quench the fluorescence of the fluorophore. Now, this vesicle is special because in the membranes, we have gramicidin A, which under kind of normal states with this system, it exists as a separated dimer that really doesn't allow thallium ions to go through efficiently. But what Olaf has shown through his whole career is that when you add a membrane bilayer perturbing agent, it alters the membrane physics, it bends and compresses, it forces that gramicidin A channel to form a complete dimer, and then the thallium ions can enter much more efficiently and quench the fluorescence. So then if you have an assay that measures the rate of quenching, you have a surrogate measure of membrane perturbation. So click. When we look at ivermectin, you can see at nanomolar concentrations, it's already significantly perturbing the membrane physics, right? And you contrast that with remdesivir, which only does so at concentrations like a log or two higher than its EC50. So along with some other data that we're hoping to submit soon, we think we have a new explanation for the in vitro effects of ivermectin in the SARS-CoV-2 repurposing assays. And hopefully the lessons learned from this will, one, we use to more efficiently do repurposing studies, and also hopefully provide some closure on kind of what we've all witnessed. And so look forward to that in the future. So click. So just in summary, with ivermectin, click. The antiviral activity highly correlates with host cell injury. So absolutely in vitro, ivermectin decreases viral burden, but it also decreases host cell viability. Next. in repurposing, I think this shows that clues to nuisance behavior can be provided by a classical drug mechanism of action. In this case, it's membrane-based activity. Next. That nonspecific membrane perturbation is probably an underappreciated source of cellular nuisance behavior. So the key lessons from this are learn the characteristic readouts in your assay of what these compounds can do. And in this particular SARS-CoV-2 assay, it's cytotoxicity. And places doing a lot of these assays, cellular assays, think about establishing a counter screen like the one that Olaf showed you, or a selectivity panel that focuses on membrane targets. Next. And finally, why I mentioned the AlphaScreen behavior is that you could have interference in a biochemical assay, and then when you go to a cellular assay, you can have a different kind of interference. And that leads us to the point of you have to be very careful about associating a cellular readout with a target-based readout. You can't just say it's active against my target, it's active in cells, therefore that's because of this specific mechanism. You need a lot more data to make that connection. Next. So now I want to finish up with some of the tools for addressing nuisance compounds. Click. I'm not going to belabor this, but things like cellular health assays and kind of best practices for aggregators and electrophiles, they're outlined in the assay guidance manual. We're going to hopefully get a chapter out soon about high content screen artifacts. And then kind of the design of the overall workflow and the philosophies I kind of discussed here, I would go to this Cell Chem Bio paper published last year. And then the final thing I want to talk about next is the use of informer sets with reference nuisance compounds. Click. So this is something that I really advocate strongly now for, where in our cell painting assay, for example, if you were to take a select number of prototype nuisance compounds, like the one shown on the left, you could, in theory, craft coverage of all of those cellular injury phenotypes associated with gross injury, tubulin poisons, et cetera. And that's important for the following examples. Let's say you're doing assay development work and you use an informer set like this, where you use increasing concentrations of electrophiles shown in the upper right. If you had compounds, if you found out that at the higher concentrations where you saw kind of those dead looking cells, you might want to change your screen and say, maybe we should screen at a lower concentration so we're not enriching for, super hot electrophiles, right? And it's not just you're doing a thought exercise, you're getting the data to justify that decision. The other is in hit prioritization, where if you can map what the particular phenotypes that are associated with each of these mechanisms that are undesirable, when you see a compound that produces that same phenotype, you can flag it and say, maybe we should go elsewhere, or we should do some follow-up experiments. And click. The biggest selling point for this is that if you create such a set, you could use it across any biology or assay. You don't have to create a fit for purpose one of these or counter screens for every single assay that you're doing. So a place that does a lot of assays, for example, like the national labs, this might be something useful. So click. And there's already a more extensive published set in that Cell Chem Bio paper shown here. If you go to the SI, there's several hundred compound list of technology, non-technology, and cytotoxic compounds, including active and inactive analogs and multiple chemotypes for each one. So you can really be more confident that any readout you're seeing is associated or not associated with one of these nuisance compounds. So this actually lets you generate real data and to see, because like I mentioned at the beginning of the presentation, it's really hard to predict how a cellular readout is going to be with any one of these mechanisms. You really need to get that data. And so I can tell you, click, that one big pharma already basically took this and assembled their set internally. And the feedback we got already that I got was that it already changed how they're performing screening. I will also tell you that this is an effort of a consortium that I helped establish, where they're trying to basically make a reference library to be used by everyone. And I think the best contact, if you're interested, would be Jared here at AstraZeneca. And this is still going on. And I think we're at the stage now where they're looking at like commercial partners to help build this and work on how to disseminate this at a reasonable cost so everybody can use it. Okay. And so finally, to conclude, my top advices for cellular nuisance compounds. One, thoroughly characterize the effect of your compounds on cellular health, the full response curve, time courses, and multiple methods. I think that Ivermectin data shows the importance of that. Next, determine the effect of nuisance compounds on your assay readout. Again, that you can do that with the set of informer compounds like I just showed you. Try to get evidence. How easy is it to get a positive readout? If you're doing a loss of signal, like in a lot of these anti-cancer assays, you'd be surprised at how easy it can be. Next. You should counter screen in cells with unrelated, and if you are able to generate like a target null cell line, that's always highly recommended. And then finally, you really want to be aware of a confirmation bias when connecting a target-based readout to a cellular assay. And I think, again, showing you that just because you see activity in, say, a target-based AlphaScreen, and then you put it in cells and you see some antiviral activity, it may not always be as simple as that. And so with that, I just want to acknowledge a whole lot of people. The cell painting paper has, I think, like 30 authors on it. So I didn't list them all, but there's a ton of people that did it. With this work at NCATS and doing the Ivermectin work, here's some of the key people, as well as other work with the assay guidance manual and in other data that I showed here. So with that, I'm happy to take questions.
Jayme, that was fantastic. I'd like to open the floor for questions, please.
Yeah, Jayme, this is TC. Fantastic talk, really, really eloquent. And I had to step in and out, so I might have missed something. But I had two questions. One is with regard to these nuisance compounds and nonspecific membrane perturbations. Is that mostly seen in live cell assays, or do these effects disappear if you make membrane preps from lysed cells?
Yep. Anything where there's a membrane, because it's going to essentially, these compounds, they sequester or enrich in membranes. So the first PDSB profiling, those were radiolabeled just hot purified GPCR extracts, right, or whatever.
I see.
And
so not intact live cells, anytime they're membrane associated. So even with nanodiscs or something like that, membrane particles.
I haven't tested nanodisc, but in principle, yeah.
Okay. And the second question relates to your cell painting with electrophiles. What have you actually deliberately looked at these profiles in terms of time dependence, like if you incubate it at shorter times versus longer times?
So we did an experiment at 24 versus 48 hours. So you're a little limited because of the throughput and all the rigmarole of doing these big screens, right? We didn't see much of a difference between 24 and 48. And that in that paper that we have, some supplemental data and figures to show there really isn't much of a difference. If it's really a hot electrophile, you'll see it at 24 hours.
Yeah.
But then you could imagine on a smaller scale, if you're only working with one plate, then doing a 6 hour, you could conceivably do.
I see. Okay, so that's something you could actually test empirically.
Yep.
Okay, great. Thank you. Great talk.
Thank you.
Jayme, you've studied nuisance compounds extensively and pointed out that sometimes you can have nuisance behaviors in your biochemical assay and then your cell-based assay. You showed that nice Pfizer style diagram of the complexity of cell-based assays, right, which are typically substantially more complex than biochemical assays. Do you have a sense of the prevalence of in the context of high throughput screening, where folks will be screening hundreds of thousands of compounds from, combinatorial libraries, a sense of kind of what the nuisance hit rate would be for, in general, biochemical versus cell-based?
Well, I remember one talk where basically it was a biochemical assay and they did the 3 sigma line, right, for hits. And, there's like 1,000 things above. And basically the presenter basically said, so everything above this line, 99% of it is not, is probably some sort of artifact or interference, right? I don't know if it's quite that high, but I think we all know from kind of doing follow-up that, if it was that easy to find a real compound and that was not acting by an undesirable mechanism, we'd have a lot more leads, right? I think the safe bet would say double-digit percentage, and some, if you have like a cell-based assay where you're kind of measuring, let's say like it's impacted by cellular damage, like an antiviral assay, I would say you're probably, the majority of the things are just going to be cytotoxins.
Mm-hmm.
And you'll find with different targets have different sensitivity profiles to nuisance mechanisms. So for example, a compound that has kind of a cysteine switch on it, you can imagine that is going to be ultra sensitive to electrophiles, right? But then let's say there's a protein that doesn't really have any nucleophilic residues on it. That's probably going to be more resistant to modification by an electrophile, right? And so that's one of the kind of the biggest problems with when people invoke selectivity at the early stage is they'll see activity in one and do a counter screen versus a different protein, and they'll see a apparent difference in the EC50 or the IC50 values. But what they're not controlling for is, well, is that other target that has the same sensitivity to nuisance behavior, if that makes sense.
Yeah.
So for example, there are compounds or enzymes that, like AMP-C, that are super sensitive to aggregators. And then there are other enzymes that you can throw every aggregator that you possibly know, and it doesn't do anything to the target. It's just not sensitive to it. And so if you were to use that as a counter screen in an aggregator, you'd say, oh, my compound's selective.
Yeah.
Yeah, Jayme, that just says you have to really be careful and know your system. I mean, that even is compounded when you're trying to SAR and have selectivity, because you'll have different levels of flatness depending upon the target, right?
Exactly. And
So you can fool yourself that you have an SAR.
Yeah, so I think what the advice I got in grad school, I think the best advice is do the experiments, right? And even these, well, why, this is a lot of work, or do we really need to do that? Well, a lot of the data I showed you is not that, it's not a fancy type of assay. We're talking about cellular health assays, doing appropriate controls, et cetera. So I think it can be done.
That's great.
Any other questions?
Hey, Jayme, I got a question, if there's time.
There's
Hey,
time.
Johnny.
Hey, so from your perspective, so I mean, I love the informer set. We've taken that to heart, and we routinely screen an informer set that was basically that, it was advice that you gave us in all of our screening campaigns. What's your thoughts on the ability for machine learning to be able to predict this from physiochemical properties? I mean, informer set is great, but do you think we can use like physiochemical properties to basically predict, compound promiscuity in cellular-based assays? Do you think there's enough information in the chemical structure? Like certainly where the membrane associating compounds, those probably all look similar, right? But just curious about your thoughts of if that would work or not.
I think it can. I think the problem is depending on the data you're looking at, right? If it's a biased set or how, I think the more confidence you would have in such a method was you'd really want to train that on a very good reference set itself. But there's, this kind of relates to the whole pains, no pains. What's the utility of using flags like pains? But then with cellular assays, there's so many different biologies and assays you can test that's kind of why I've been pushing more lately on the informer set, because you actually just generate the data itself with every single assay that you do, and you don't have to worry about, was this applicable, this training set applicable to my assay. But at the same time, I think there are things where it's highly insoluble or highly lipophilic. Well, yeah, there probably is going to be some, I don't know what your ROC curve will be, if it's going to be 80% and 80%, right? Is that going to be useful for making a major decision, or would you just want to generate the data? I think ultimately, I think the safe answer is probably it's important to have both, especially when you're talking about screening large libraries. And if you're going to be a place that's routinely doing screening, it may be worth doing.
Yeah, I agree, but it would be great to have, from the chemical structure alone, it would be great to have some insight into the mechanism of the non-specific activity, like at least to narrow the hypothesis field and to point you in a specific direction. Yeah.
Oh yeah, and I think that would be great. There are, for a lot of the cell-free based assays, there's a lot of data sets from places like MCATs. Those data sets are out there that, are still being mined today for that. I think the challenge is where, especially when I did my postdoc, is how do we then take the next step to cellular assays? Because that to me is kind of an unsolved problem still.
Yeah. Great. Thanks.
Because kind of all bets are off. I mean, it's a black box, right?
That's for sure.
Yeah. All right. Any other questions? Jayme,
I just say,
thanks
if you have
again.
questions, my email address is at the front, so feel free to reach out.
Great. Thank you again. Thanks, everyone.
All right.
Goodbye.
Thanks, everybody.